Everyone, welcome. We're so glad that you're here. And I wanted to point out that we have a sign-up sheet over there. This is our first seminar of the John S. Gates series, and we just wanted to see how many people would come from various parts of the state to be here with us, and of course, virtually. So I would like to introduce Dennis Roop, who is here to introduce the seminar series as well as our speaker. Thank you.
Thanks, Jill. As Jill said, I have the great privilege of actually holding the John S. Gates Endowed Chair. John and his sister, Diane Gates Wallach, made the initial gift that established the Gates Center in 2007. And then again, in 2022, they made a very generous gift that enabled the establishment of the Gates Institute. So John was trained as an architect, and as a result, he was very curious about the design of things and how they worked. I remember when John first toured the Gates Center back in 2008, we took him on a tour of the cell sorting facility and showed him the new sorter that we just purchased. John spent about an hour questioning Karen Helm, who many of you may remember was the director of the facility at that time, about the design of the sorter and how it worked. Next slide, I think. Oh. Maybe. Okay, there it is. So this slide was taken during John's first tour of the Gates biomanufacturing facility. He was accompanied by his wife Jules on the your far right, and Xiaojing Wang, who actually was the first holder of the John Gates Endowed Chair. As Matt Seefeld, I don't know whether Matt's present or on the call, but as he can attest, Matt is the director of the Gates biomanufacturing facility. John spent a lot of time in the facility asking lots of questions about how things were designed and how they worked. And I think in the end was really amazed at the capabilities of the GBF. Unfortunately, John passed away last year. And when we were discussing with Diane how we might acknowledge and honor John's unwavering support for the Gates Center and the Institute, she thought that rather than fitting or placing his name on a plaque and hanging it on a wall, it would be more fitting to honor his lifelong curiosity and support by permanently naming the Gates Institute Seminar Series the John S. Gates Seminar Series. So today, I'm very pleased to introduce the inaugural speaker for this seminar series, Dr. Mike Verneris. Mike holds the Barton Endowed Chair of Pediatric Bone Marrow Transplant and leads Translational Sciences at the Gates Institute. The title of this talk, I think, is on the next slide, Gut Instincts, Translational Studies of Stem Cell-Derived Innate Lymphoid Cells for the treatment of GI graft versus host disease and Crohn's disease. Mike, the floor is yours.
Thank you very much. Can everybody hear me okay? Or?
Yes.
Good. Thank you very much, Dennis. And thanks to the Gates family for really sponsoring this lecture series and all the incredible support they've given us at the Gates Institute. It's been an incredible journey thus far. I thought I would kind of talk about a little journey that I've been on for a long time. That really starts pretty much at the beginning of my career. And it started in a way that it doesn't end. And, one of the things I don't like in medicine is lifetime achievement awards. Because lifetime achievement awards are, I was a medical student and saw this rare disease. I was interested in those patients. I cloned the gene. I made a new protein. I treated them, and it was all perfect. And that's the way it goes. But this is a story about a long and winding road and moves in all sorts of different directions. But it kind of started when I left Stanford University and moved to the University of Minnesota in 2002. And at that time, in 2002, was the first reports of using haploidentical NK cell infusions for the treatment of refractory leukemia. And so when I got there, I had just been doing work with T cells for the last 10 years, and I wanted to move over to NK cells to try and, kind of capitalize on some of these early discoveries. And so I was working, I don't know if this works or not, I guess not. I was working with mature NK cells and trying to make them proliferate and expand so we could treat patients with refractory leukemia. And after about a year of doing this, I learned that NK cells don't grow very well in the lab, or at least they didn't at this point. They expand 1 or 2-fold at most, but you really didn't get many more cells to treat patients. It was clear that we probably needed more cells. So I went to my mentors at that time and I said, I just read this paper about how this guy, Tucker Libyan, who's here at University of Minnesota, is taking hematopoietic stem cells and turning them into B cells. And can I do this with NK cells? And people said, sure, give it a try. And so I, after 2 years of trying, developed a system where we took CD34 cells, and again, this was for patients, and this proved how naive I was at that time. I took a mouse fetal liver cell line and grew the CD34 cells on that. Little did we know that the FDA would not have liked that very much. Anyway, we took the CD34 cells, we grew them in a bunch of cytokines, we got these cells we called NK progenitors, and then after about 4 weeks, we got large numbers, about 3,000-fold per 1 input cell of NK cells that were cytotoxic in some cases, made cytokines, had the right receptors, and all that kind of stuff. And we published a whole bunch of papers on how this worked and how to do this, et cetera, et cetera. And then in 2010, there were 3 papers that were published. One by Mike Caligiuri, the top one that was in blood, and he was working in a very similar system to me, although he was looking at lymph nodes and looking how NK cells developed in lymph nodes. And the second paper was by Marco Crohn in Nature, and the third one was by Jürgen Spitz in Nature Immunology, and Jürgen was in the Netherlands. And so these 3 papers all describe an NK cell-like cell that made interleukin-22. And so these cells were different than regular NK cells. They didn't exist in the blood very much. They're extremely rare. They couldn't even find them at this point. They lived in the tissues. Caligiuri found them in the lymph nodes. Marco Colonna found them in the intestinal lamina propria, and Jürgen Spitz found them in aborted human tissue. And that they were thought to be an NK subtype, thought to only exist in lymphoid tissues, and they had this unique cytokine production, as I say, called IL-22. And they expressed this transcription factor called ROR gamma T. That's all we knew. They were interesting. And so then I went back to shortly after their report, I went back to our system because some of our NK cells never really developed in the way they should have. And lo and behold, what I found was that we too were making these same NK22 cells, kind of shown here, this CD117 high population. I won't go into the details except to say that production of interferon, which was the kind of hallmark cytokine of NK cells, was mutually exclusive for productive of IL-22. So these are clearly different cells, and there's a lot of question in the literature about what they were. And now over the last 10, 12 years, there's now at least 6,000 papers about a cell type called innate lymphoid cell. And so really these early descriptions were of ILCs, or innate lymphoid cells. So this is the 4th lymphocyte described in humans and in mice and in all mammals. And so there's B cells, there's T cells, there's NK cells, and there's ILCs. And so we were, in addition to making NK cells, making ILCs. And if you look, we and many others contributed to this figure where you kind of start with a hematopoietic stem cell, and all of those boxes are various transcription factors and cytokine exposure. And if you go to the left, you end up becoming a T cell, and if you go to the right, you end up becoming an ILC. And hopefully you can appreciate from this figure that they are really quite mirrors of one another. In other words, in that whatever that darker colored box at the very bottom of the chart, on the left-hand side, the T cell, that's a CTL or a cytotoxic T cell, essentially mirrors an NK cell. And a Th1 helper T cell mirrors an ILC1, and the Th2 mirrors an ILC2. And again, these cells mirror one another, and the difference really is the T cells have a T cell receptor, whereas the ILCs don't have a T cell receptor. So evolutionarily, these are more immature cells and more innate lymphoid cells, and that's where the name comes from. And really what they do is they respond to tissue signals that basically lead to tissue homeostasis. And when homeostasis is breached, they become active. And so again, so in the type 1 ILCs, basically these cells respond to IL-12 and IL-15 and IL-18, and they then become activated and either develop cytotoxicity in the case of an NK cell, or they develop cytokine production that leads to other aspects of the adaptive immune system responding. In the type 2 cells, again, they're more like worms and helminths, and those organisms, those parasites basically lead to epithelial disruption and injury, and then expression of the alarmins, which are IL-25 and IL-33, which activate the ILCs to make IL-4 and IL-13 and IL-5, which leads to worm expulsion, or AREG, which is an EGF protein that leads to epithelial regeneration. And then the type 3 cells also respond to bacteria, and usually dendritic cell-derived cytokines such as IL-1 beta and IL-23. And these cells in turn make IL-17, which is a very inflammatory cytokine, or IL-22, which is a restorative cytokine, which basically acts on tissue stem cells, either in the skin or the GI tract mainly, and lead to restoration. So if you have too much IL-22, for instance, you develop psoriasis. If you have not enough IL-22, you can get GI tract infections. And so this is kind of how ILCs work, and they've been implicated in a variety of diseases. Many allergic diseases ILCs are implicated in. Fibrotic diseases ILCs are implicated in. Crohn's disease or inflammatory diseases, and obesity. And it's really more of like a yin and a yang. In other words, if you have an imbalance of ILCs, then you lead to problems. So the balance leads to protective barriers, less viral production, better viral clearance, and basically tissue homeostasis. And if it's imbalanced, then it's the opposite. Basically, it's epithelial barrier injury and infections and hindered wound repair. And so I'll leave some of the kind of the nitty-gritty stuff out, but armed with all this information and knowing that we had this system, I mean, really, when people were first trying to study ILCs, they were basically like, killing hundreds of mice to try and get enough cells out of the tissues, or they were using aborted human tissue, or trying to get pieces of GI tract that were removed for various types of surgeries to get these. So very hard to study. And we had this nice system that I showed before just to generate literally hundreds of thousands of them. And so work that we did in our lab more recently kind of identified which progenitors give rise to either NK cells shown in the red box or ILCs in the blue, in the mustard color box there. And we started to understand, can we actually make some of these in the lab, and what do they look like and what do they do? And I won't go into the details, but we spent a lot of time, and this is a paper that we published in Science Immunology. It probably had about 100 figures all together showing that these cells actually do what their naturally occurring counterparts do. So all the events that we were kind of doing in the dish seem to recapitulate the behavior of the cells in the human or in the mouse. And in fact, when we injected these into immune deficient mice, you can see in the top panel up on the y-axis is a human CD45, and on the bottom is the mouse CD45. So looking at human cells in that top upper left corner, you can see when we injected the cells that should give rise to NK cells, they only ended up in the bone marrow. However, when we injected these ILCs, they ended up in the tissues. They ended up in the bone marrow and the spleen, but they also ended up in the liver and the lung and the GI tract. And so again, we really kind of felt like we were kind of on the right track. And so we patented some of this stuff, and we met with a couple of venture capitalists, and they thought, great, but what do you got? What do they do? Like, do these cells do anything? And we were kind of stymied because we never thought about that. And so really they asked us, do these cells work? Prove to us, and then come back to us. And so in fact, that's what we've been spending the last probably 3 years doing is basically taking blood or taking cord blood or bone marrow, trying to isolate the stem cells, turning them into ILC2s or ILC3s or combinations, and then using mouse models and trying to understand how they work. And so just to kind of digress for a little bit, these ILC3s especially live in the intestine. And this is kind of a cartoon, like many cartoons and many review papers, but you should get this look at this ILC3. It lives in the center of this figure because it's about how ILC kind of promote intestinal health. But you can see that this in the light blue area on the epithelial border, there's this mucus that our intestines have, and there's bacteria. And then in our diet, we have these AHR ligands that basically go through the intestine and act on the ILCs to keep them healthy and keep them growing. And these ILCs, in turn, there are T cells in the intestine, and they present bacterial antigen to those T cells and make those T cells quiet so that they're not causing problems. And they do a whole bunch of other things, kind of feeding back down here to the goblet cell and the Paneth cell, and keep the intestinal integrity alive and appropriate. However, in Crohn's disease, for whatever reason, you end up with this epithelial injury or this epithelial dysfunction, and then the mucous membrane's less. The bacteria kind of traverse into the lumen of the intestinal wall and create inflammatory signals, and this disruption of ILC function, where these ILC3s, which are the restorative healthy ILCs, now start to become inflammatory ones and driving inflammation and tissue injury. And it's this kind of cyclical problem that happens in Crohn's disease. And there have been many studies now that have looked at people with Crohn's disease and shown that their ILC3s are depleted, either because they just simply die or they become inflammatory ILC1s. And I think it's an adaptive response that happens in normal health, but it's exaggerated somehow in Crohn's disease for a variety of reasons. And by the way, I'm not an all Crohn's expert, but this is what I've gathered from the literature and like kind of looking at this. So again, we start asking questions: can stem cell derived ILCs be used to treat GI tract inflammatory diseases? And the challenge here is that these cells are tissue resident. Like, there's no way that I can harvest cells from you guys. And the mouse studies that have looked at this, the donor mouse doesn't end up doing so well. They die. And so it's really not translatable, clinically translatable. And so our hypothesis was that these stem cell derived ILCs might be able to be used for treatment of these GI tract inflammatory disorders. And so I kind of bugged Ed Zoten in pediatric GI about 1,000 times and asked him about models and what we could do. And he kind of shared with us a mouse model that he was using called TNF delta ARE. And I'm pretty sure that TNF delta ARE is a mouse that's basically been engineered to have a longer poly-A mRNA, which increases stability for the TNF molecule, which is implicated in the pathogenesis of Crohn's disease. And so these mice as you like in the bar graph here, you look at the inflammation in the intestine, in the duodenum and the jejunum, it's relatively low, but in the ileum, it's quite high and inflamed, and in the colon as well, it's not. And that's exactly what happens in humans with Crohn's disease. They get an ileitis. And if you look at the next picture, it's a wild type mouse on top and a Crohn's disease mouse or a TNF delta ARE mouse on the bottom. And anybody who's worked with these mice can attest to their fragility, and they're just really sick animals. They're just not well. You can see that in the picture. And then the bottom is the GI tract, the ileum of these mice at 4, 8, and 20 weeks. And what you should kind of appreciate is that villi is kind of blunted. There's a lot of purple in there, which are lymphocytes. And then it's really thick in edematous, and they're super sick by the age of 20. And I think these mice also develop other manifestations of Crohn's, like arthritis and things like this, which we haven't touched in our work. But really what we asked was, could these ILCs, these stem cell derived ILCs, be used to treat these mice? And so if you look at a wild type mouse and you feed them for, whatever, you just let them have a good, happy life, like humans, they gain weight. And if you take these TNF mice, they tend not to gain weight, and they even lose weight. And so the hypothesis was that if we added these ILCs, maybe they would gain weight as an indication for their health status. And this graph is basically showing that. So on the y-axis is the percent weight gain over time. And you can see with those blue arrows on the x-axis, we treated the mice with weekly infusions for 3 infusions at week 6, 7, and 8. And then shown in the line graphs is the black line, which is just a wild type mouse that just gains weight over time. And the purple line is the TNF mice that just got treated with PBS. So you see there's quite a bit of difference. They're all runted and not doing so well. And their weight gain plateaus, from week 12 onward. However, in the red line where we treated the mice with the ILCs, you see a little separation of the curves at like weeks 9, 10, 11. And then we said, well, why don't we treat them again at weeks 14, 15, and 16? And you can see when we treated them at weeks 14, 15, and 16, again, we kind of cut down on some of that inflammation. And then when we added a second dose, we tended to really stabilize that growth and that weight gain of these mice. And just to kind of look at, you could gather further information about this. And by the way, I'll say this is definitely work in progress. Like, this is not a complete project yet. Dejeni basically gavaged these mice with fitsidextran, kind of shown here. Basically, there's a fluorescent dye that you put into their stomach, and it goes through their intestinal tract. And then you can look in their blood later, 24 hours later, I think it is. 24 hours? 8, whatever, 8 hours later, and you look in their blood and notice that I've been doing all the experiments. But you look in their blood for the fluorescent material, and if the fluorescent material is there, then their GI tract is not very healthy, and the dextran is leaking into the blood essentially. So if you look at these graphs, either after the first treatment or the second treatment, you see that the wild type mice all the way to the right have a relatively low amount of fluorescent material in their blood. The ILCs treated mice with TNF are very similar. The mice that got PBS, on the other hand, are significantly higher, meaning that we are actually impacting the patency of the intestinal mucosa, which I think is pretty cool. The other thing is I just added this at the last minute, but we're just to simulate what this might look like in a human, we took the TNF overexpressing mice and made ILCs out of those mice and treated them either with PBS, shown in purple, or treated them with their own ILCs, shown in red. And you can see that their weight gain is just as good as with wild type ILCs. So in other words, even if we make ILCs from affected people, this may still work. And are there changes in the microbiome? Yes, there are changes in the microbiome of these mice, but unfortunately I don't have the data put up here. We've looked at the raw data. There are definitely changes. We need more mice, but we're definitely seeing some changes in the microbiome. So kind of moving on to the other disease model that we used, I'm a bone marrow transplant doc, and bone marrow transplants curative for diseases that aren't responsive to other conventional chemotherapy. It functions through the high doses of chemo and the graft versus leukemia reactions, and it's a high-risk procedure with about 25% of patients dying in the first 100 days. So it's pretty profound treatment. Higher risk than brain or heart surgery. And if you look at it, patients get high doses of chemo and radiation. We collect either bone marrow or peripheral blood or cord blood, and then we infuse them into the patients and hope for the best. But one of the major side effects of bone marrow transplant is graft versus host disease, is those donor cells that aren't tolerant to the patient recognizing target tissues such as the skin in the upper two panels, or the GI tract in the lower two panels, or the liver in the bottom panels as being foreign and attacks those tissues. And it happens in probably about 30–40% of patients. The majority of patients can be successfully treated, but it is a life-threatening complication. It definitely has an associated mortality with it. And similar to Crohn's disease, at least GI-GVH, which is the most severe form of GVH, really is an immune-mediated attack on the intestine, and it's very similar in terms of the donor T cells recognizing antigens in the patient's GI tract and attacking and causing a breach of mucosal barrier and translocation of bacteria and inflammation. And prior studies have shown that, I said that the ILCs are almost never in the blood, and they're in the blood, but at like probably 0.001% of all the lymphocytes, extraordinarily rare. But this study performed in the Netherlands looked at patients prior to transplant and looked at ILC1s, ILC2s, and two different types of ILC3s. And they said if these cells were active, now active by CD69 expression, and then they looked at patients above or below the median for those three groups. And you can see in the bottom Kaplan-Meier curves that if you had high numbers of activated ILCs, you were much less likely to get GVH. And that's pretty interesting, right? And there's other mouse data that supports the role of ILCs in a protective, GVHD protective manner. And so to model this, again, we took our human ILCs and immune deficient mice. So we had 4 groups, one where the mice got blood from just a healthy donor that would cause GVH in this mouse, a second group that got blood in the ILC2s, a third group that got blood in the ILC3s, and a fourth group that got both ILC2s and ILC3s, but no GVHD provoking cells. And we treated them 4 different time points, 0, 7, 14, 21 days after transplant, and we looked at the mice. And so again, here's the survival. In the gray line, you can see the mice that just get PBMCs and PBS all die of GVH in about 35 days. However, the mice that get either the PBMCs and ILC2s or ILC3s, they are protected and live longer. And the mice that just get ILC2s and ILC3s and don't get GVH do just fine. And shown in the bottom is the GVHD score, which looks at how the mice walks, their fur, their behavior, their diarrhea or not. You can see the ones that have high score basically don't survive and die quicker. And so again, kind of suggesting ILCs do something in this model. And then we went to a pure mouse model, which is fully mismatched. It's like me and to any of you guys. So no matching, no nothing. And so in this model, we take C57 black 6 mice and transplant bone marrow into biopsy. And if you transplant the mice with just PBS, they don't get GVH. If you add splenocytes, they get GVH. And then we tested either donor or recipient ILCs from either the black 6 or the biopsy mice. And similar to the other studies, we see that the ILC-treated mice, either in the mustard color or the green color, are protected. They're not all protected, but they're at least partially protected, again, suggesting that these ILCs are doing something in this model to protect those mice from GVHD. And again, the GVHD score is shown down at the bottom. So we started kind of asking ourselves, how do these ILCs prevent T cell driven inflammation? And it was really, I mean, we went back and forth, we really didn't know where to begin. And so Dejani started looking at the mice and looking at the number of T cells in either the blood in the far left, or in the lung, or the liver, or the gut. And the liver and the gut are the GI target tissues. And then we looked in the groups that were treated with just PBS, or the ones that were treated with ILC2s or ILC3s. And what you should appreciate is that in the target tissues, the liver and the gut, the third and the fourth panels, basically have fewer T cells. They're not expanding in the tissues where they should be expanding and causing GVHD. So some kind of, prevention of either the cells getting in there or the cells expanding. And just to kind of explain this further, we looked at the large intestine on the left-hand side and the liver on the right-hand side, and we looked at CD3 for the T cells or CD127 for the ILCs. And you can look at the top panels and you can see those are controls. They're mice that didn't get either T cells or ILCs, and there's no staining. In the second panel down, there's the mice that just got the T cells, and you can see there's T cell staining both in the large intestine and the CD3 columns of the liver and the large intestine. And mice that got the T cells and the ILC2s or the T cells and the ILC3s, you can see the ILCs are there in both tissues, but there's very few T cells. So again, it corroborates kind of what you've seen before. And not all T cells are the same in terms of causing GVH or causing inflammation. And it turns out that the naive T cells, the N-like cells in this panel, are the ones that really cause GVH. And those cells, if you look, those are the ones that are depleted in ILC2 and ILC2 and ILC3 treated mice, whereas the other T cell subtypes are really not perturbed at all. And then we started asking about what happens in vitro. Does this happen in vitro? And so, do you need to have contact, or could this be through transwell, meaning soluble mediators? And so we used in the top contact where the ILC is shown in these little squares, or the T cells in the circles were allowed to touch one another, or in the bottom in the transwell where they weren't allowed to touch one another. And if you let your eye go to the contact and the transwell bar graphs, you can see they look pretty similar. And so really what that suggests is that it's a soluble molecule that's preventing these T cells from expanding. And then lastly, we kind of looked to another way where we took and stained T cells with a membrane dye called cell trace or CSFE. And then the T cells were stimulated with 328 beads to drive proliferation. We washed away that stimulus and we added ILCs, and then we faxed the cells 7 days later. And if you look on the x-axis of these two plots, the CTV on the x-axis, the more to the right that is, the brighter fluorescent they are, the less they've divided. And you can now look in this little T to ILC ratio, and you can see that when ILCs are added at large numbers, it prevents those T cells from dividing, both the 4s and the 8s, and using ILC2s or using ILC3s. So both conditions tend to inhibit T cell proliferation. And then lastly, we use that same method to look at how ILCs activate T cells. And again, we find that when you add the ILCs, somewhat paradoxically, they activate them, right? They basically make them make granzyme, make them make perforin. So they're actually more active. And so it's this kind of weird kind of picture. Oh, and then one more thing, which is that they make a lot of different cytokines. This is data that's kind of reduced into a heat map just for simplicity. But you can see when you add the ILC2s and the ILC3s to the T cells, they're making much more cytokines than just the T cells alone. And again, so it's this paradoxical kind of effect these ILCs have on T cells, where there's fewer cells in the target tissues, there's less naive cells, the proliferation is inhibited, but the cells the T cells that are there seem to be active and making lots of cytokines. So we started looking into the literature, and there's this concept called senescence. And this is a picture of senescence, and it's just a cartoon that was convenient to show, but senescence and exhaustion are different. And I'm not entirely sure if I can explain how they're different, and different papers describe them differently, but they're accepted to be different. And in general, senescent cells proliferate less. They rely on different metabolic features, which I'm not going to really talk about, make lots of cytokines, and express particular surface receptors. Exhausted cells express different receptors, somewhat overlapping, but make less cytokines, and again, they're less proliferative. So we started looking at senescent markers, and not to belabor all this data, it's a busy slide, but again, if you compare the gray to the purple or the bluish color, you see that the purple and bluish color are high, meaning that these senescent markers are increased when the T cells are cultured with the ILC2s or the ILC3s. We also kind of started looking at the more more intracellular pathways that drive senescence. And so at least one of the accepted methods that senescence occurs is through a DNA damage response, and that this in turn activates ATM and ATR, and then turns off a variety of different proteins, which yet turn off another cascade of proteins, which then turn cells off. So if they're senescent, this doesn't happen. And so as you can see, in the Western blots, or the quantification of the Western blots, when you add ILC2s or ILC3s, we see increases in P16, which leads to cell cycle arrest, or P21, which leads to cell cycle arrest, or P53, GATA4, and FKB. B. So again, it's kind of this stimulating the cell, but causing it to not proliferate anymore. And furthermore, we kind of quantified this using immunofluorescence, where we looked at P21, and it probably doesn't project very well in this light, but you can kind of look on the right hand, on the right hand graphs, and see that there's more senescent cells when you add ILC2s and ILC3s, and still yet further more senescent cells. And so looking at cell cycle arrest, again, when we looked at a variety of different T cells, the green and the purple bars in the naive panel here, you see that they're the ones that are really been affected by senescence here when you add ILC2s and 3s. So we're pretty confident that we're inducing senescence in these cells. And so how does this happen, right? Like, how do you get senescence? And so again, it kind of goes back to those Transwell experiments that we did. It's probably a soluble molecule. So we started looking at a whole bunch of different proteins, and one stood out, which was IL-9. So IL-9 was turned on in when ILC2s and 3s were activated, but not when NK cells were. And then we said, okay, let's take ILCs out of this whole picture and just add IL-9. And when we added IL-9, you can see in these pink pictures, IL-9, when you add them, reduces T cell proliferation and reduces CD4 and CD8 T cell proliferation. Conversely, when you added ILC2s or ILC3s and you blocked IL-9, now the cells start to proliferate and you get even more cells. So you're getting rid of that senescence, is kind of how we're thinking about this. And then IL-9 again affects the T cell subsets, but mostly affects that naive T cell subset, the CD8 naive T cells on the last plot. And again, when we add IL-9, it makes the cells make cytokines. So again, it activates the cells, but it doesn't let them proliferate. So again, senescence. And then even further cytokines, which I won't go through. And IL-9 induces the senescent markers that I showed before, more senescent markers. And so really we think that what we found is that when we add ILCs, they induce T cell senescence and reduce inflammation. So now we're starting to think about how to scale this up into like a GMP facility. And so this is really the early work that Dejeni has been doing, looking at the number of cells on the x-axis, the number of ILCs we get out of our cultures in the starting material, in the starting number. This is a 24-well G-Rex plate that's GMP compliant. And if you back calculate all of these numbers, we think that from 1 umbilical cord blood and probably about 2 million CD34 cells. Now you can probably get more than that in GMP, like really good cords. You can get about 400 or about 320 million ILCs from that. Probably enough to treat a human. And so I think our vision really is that you could start either with a umbilical cord for off the shelf, or you could start with patient-derived. You would isolate the CD34 cells. You would then isolate the alpha-4 beta-7 cells. I didn't mention it, but we got rid of the fetal liver cells long ago. And at the end of about a month, you could have mature ILCs, which you could treat patients with. So I'll kind of end there and say that stem cell-derived ILCs seem to function in animal models of GI tract inflammation. They home to the tissues and they drive restorative functions. They quell inflammatory reactions through restraining pathological T cells through induction of senescence. In this model, the senescence is driven by ILC-derived IL-9. And I think that we can produce ILCs in quantities needed to treat patients. So Dejeni's back there. He's the guy who really has stuck with this project and really knocked away at it really hard, but it's been a lab effort. And literally everybody on this, on the left-hand side of the panel has participated in one way or another in this research. I want to thank Ed DeZoten. I don't know where, I guess I forgot to put the guy's name from Ed's lab. I put Dejeni's name instead. Tom Nguyen. And I'd like to, some of this work was funded by the NIH, and I'd like to thank Gates Grub State for really supporting this work, and I'll stop there and take any questions.